thank you very much for tuning in at this slightly unusual hour for a CDA seminar. It's been arranged at this time to accommodate Professor Mark Cooper, who is zooming in from Queensland in Australia, where the local time is 4 o'clock in the afternoon. So it gives me great pleasure to introduce Mark, who holds the chair of prediction-based crop improvement at the University of Queensland. And Mark, he's a quantitative geneticist who uses modeling and genomic prediction to inform plant breeding. And he focuses above all on drought tolerance, in particular in cereal crops such as maize and sorghum. So today's topic also leads on very nicely from the seminar we had last week by Ed Buckler. So a few words about Mark. He's one of those, let's say, dark horses who, after his PhD degree, which was also in the University of Queensland, he joined industry, and there he worked his way up through the rank and file before about 3 years ago, switching back into academia to take up his present position. So for the bulk of his career, Mark has worked in the States, above all in DuPont Pioneer, where he was part of, to use his own words from his homepage, the world's largest maize breeding program, and a testament to the practical outcome of Mark's work is not just the list of 30 patents, which is impressive in its own right, but the fact that his program led to the development and release of the so-called maize Aquamax hybrids that presently cover millions of hectares worldwide. So I first heard about Mark's story about 1.5 years ago at a meeting in Canada, and I was absolutely blown away by this impressive exemplar of the power of modern plant breeding. And so, like a little child today, I'm really looking forward to hearing the story once again. So just a few more words about Mark. Apart from studying plant water transpiration in minute detail, he also has a very deep affinity and personal experience with human perspiration. So he's an ultra marathon runner, and he competes at the very highest of levels. And he recently joined that very small, extremely selective club of elite ultra endurance runners by completing a marathon on every continent within the space of only 7 days. And in doing so, he also raised thousands of dollars for charity. So with this, I'd like to welcome Mark again to give this week's CDA seminar. And Mark, the floor, sorry, the screen, it's all yours.
Oh, okay. Thank you very much for that introduction. And I'll see if I can live up to the quality of that introduction. So what I'm going to do is share my screen first. I'll get this running, and we'll make sure that's working. So I'll put it on slideshow mode, see how we can get that going, and just do a quick check to make sure that the laser pointer is visible as well.
That's working. That's working fine, Mark.
Okay. Well, thank you very much for the really kind invitation to give a presentation. And given that I had this sort of broad topic that I was allowed to talk to, I thought what I would try and do is build on a theme that was discussed at the meeting where Brandon and I met when we were in Canada. And what I would like to do is share some ideas and thoughts on how perhaps we can rethink some of the details of the breeder's equation, which really is a workhorse of plant breeding programs that most people would have some understanding of. They may not use it as a tool actively inside their program, but they would understand how their program relates to the components of the breeder's equation. And I'll start off talking a bit about the background to that, and hopefully build from there and talk about the ideas that we're looking at that connect into the Australian Research Council Centre of Excellence for Plant Success in Nature and Agriculture that we've initiated with the main node of that, the administrative node from the University of Queensland, and there are a large number of partners within Australia and globally in this space. So I've just tried to put a bit of a gallery of a range of colleagues that are working with me in this space. Christine Beveridge, who is the director of the Centre of Excellence, Graham Hammer and Charlie Messina, who are two colleagues long-term that have been working with me in terms of trying to educate me in crop models. I sort of got a deep interest in traits and how we can use that understanding in breeding from various perspectives, whether it's through mapping or transgenic approaches, editing, so forth, but how we integrate that information. And there's a group of quantitative geneticists over here, Dean Podlick, Frank Technow, Owen Powell, that have worked with me at different times and continue to work with me on some of the background quantitative genetics theory and keep me honest in that space. And also David Jordan, who is a long-term colleague, but now we're working together to look at the ability to translate some of the work that we did in maize and what potential that has in sorghum as a translation target for the environments in Australia. And Daniel Ortiz Barrientos, who also is an investigator in the Centre of Excellence, is a connection on a lot of the adaptation and evolutionary selection trajectory. So it's sort of a connection beyond applied breeding to selection theory and adaptation in natural systems as well. So it's a bit of an introduction, and some of the work that I'm going to talk about will cut across activities with these individuals. So the other piece that I would say is that I would totally encourage anybody to ask questions as I go as well. I think that's in the spirit of this presentation. And so I'll just do a double check before I start launching into the talk that you can hear me okay, and it's okay for people to jump in and ask questions. Brand, I'll just confirm that with you.
Yeah, I can hear everything perfectly clear. Thanks.
Okay, thanks. So just as again a bit of a start, what I'm going to try and do is weave together my long-term interest in breeding and how we realize genetic improvement for a crop. And so this is a long-term study that I've been involved in, starting with Don Dubik, who used to be the director for research in Pioneer, who started this work back in 1972. And I was fortunate to start working with him on this in the late 1990s, early 2000s, where they'd been looking at successful high-impact hybrids over the history of the breeding program and how the yield had been improved in samples of environments taken from Corn Belt environments, particularly around the central Corn Belt, that I've referred to here as the target population of environments. You could also think of this as Iowa if you actually thought about it as the center of the US Corn Belt. And as Brand mentioned, I've got a real interest in how this long-term gain actually played out in water-limited environments and our opportunities to improve stress tolerance in maize and how that's played out in this long-term breeding program. So here's the same set of hybrids also evaluated in a series of environments where we managed water availability so that we could look at long-term increase in yield in water-limited environments. and then the complement control here above the well-watered favorable environments to look at what's happened in terms of improving the yield potential when we try to remove a lot of these water limitations and other sources of limitation. And so you could start to think about the potential for yield loss in terms of the difference between performance in the favorable environments or in the target environments in the on-farm conditions in the Corn Belt or in drought environments when they actually turn up here. So what I'm interested in is studying this from a quantitative genetics perspective, understanding the genetics behind this, but also how traits have played out to impact this long-term gain. And what I'm going to talk about is asking questions about this using this framework of the breeder's equation, because what I'd like to do is create sort of an ability to capture prior knowledge about traits and what's contributed to this long-term gain to enable predictions about what we might be able to do in the future going forward. And so in terms of just the specifics of the breeder's equation, which I'll come back to, you've got a selection intensity component here, this I, heritability component, some measure of variance, the amount of time that it takes to turn over a cycle, and the increment of this change in genetic improvement. The important thing just in terms of the form of this equation is to realize that the breeder's equation is a rate equation. So it's basically looking at rate of change in performance of a trait, which is sort of indicated by the slope of these lines here. And there's a number of ways you could actually measure that rate of change. What I want to do is go from thinking about genetic improvement from one cycle to what happens across multiple cycles. So short, medium, and long-term genetic improvement. And here's an example on the bottom right-hand side where I've just compared a model representing Pioneer corn breeding program with this dark line here. If we continued with phenotypic selection, this was just a simulation study that we did of how we would expect yield to be improved and by selecting on phenotype. And then we were looking at what if we could actually understand some of the genomic regions that were influencing traits that influence yield, and we could capture some genetic knowledge of those and what sort of improvement in rate of gain could we actually achieve. So you get a different slope on this line based on your ability to understand some of the gene to phenotype relationships. You can do that on a trait by trait basis. What I'm really interested in doing is integrating information from multiple traits. And the theme that I'm going to build around this gene to phenotype knowledge for traits is how we integrate that using crop growth models. And so I'm more than happy for a discussion to build around that overall theme. So that's this is my attempt at an outline slide, but not doing it in words, but trying to do it a bit by diagrams. What you'll find is that I'll focus very heavily on our ability to capture gene to phenotype knowledge about traits in terms of this additive genetic correlation component inside the breeder's equation here. If I got to move the screen around with the gallery on it. And what I'm going to do is I'm going to compare in a comparable way to these different slopes, how we use that knowledge to compare a fundamental or a foundation breeding strategies expected improvement or predictive accuracy here to some enhanced modeling approach. And can we actually get an improvement in the additive genetic correlation, which could underlie the predictions that we're making to drive genetic gain over cycles of breeding. So if both methods were exactly the same, you'd expect a one-to-one relationship. If you can come up with models that allow you to make improvements, you would expect to see positive deviations away from this one-to-one line. So that's represented here by some dots and this is a simulated example where we've actually managed to capture improved understanding of the genetics using a particular gene to phenotype modeling approach, which would result in an improved predictive accuracy, which should allow us to get a realization of an improved rate of gain if we continue that over the course of multiple cycles in a breeding program. So that's sort of the context that I'm going to try and build. Where I'll start is just to acknowledge that a lot of the ideas of the breeder's equation, where they actually began, and I'm sure a number of you might be extremely familiar with this, so I'm not going to dive into it in a lot of detail, but J. Lush has been attributed to the first formal documentation of the breeder's equation, and he was an animal quantitative geneticist breeder at Iowa State University. And he published a book, Animal Breeding Plans, the first edition, in the first version, print version, came out in 1937. It's really nice to actually go back to the pages in the book to basically see in words his formation of the ideas behind the breeder's equation, where he talked about the selection differential, S, as the difference in the mean of the selected individuals versus the mean of the original population. So it's sort of a very nice way of describing that. And he also talked about the ability to predict progeny from parent values. And he said, if you see this nice little world in here, instead of talking about this as a lot of words or this expression, he just says, for brevity, we may call the heritability of the differences which exist in the parental generation before selection began. So it sort of introduces some of these formal concepts that I think people are quite familiar with today. So tribute to J. Lush as the originator of some of these ideas. So what we can do today is sort of, we can do a lot more than what J. Lush could actually do at that time. And so I've just sort of taken the original diagram that he talked through in his book. You can actually go to something like an Introduction to Quantitative Genetics by Falconer and Mackay in 1996, and you'll find a much more detailed investigation of this concept using genetic principles and quantitative genetic principles. So what I'm going to do is I'm just going to take Lush's idea as a starting point and just do a little bit of representation of results from simulation experiments, doing exactly what Shell proposed to do, where you're looking at genetic gain as a consequence of the selection differential times heritability. We have the selection proportion that I'm going to apply is pick the top 20%. And so that'll result in a selection differential for trait values about 2 to 3% per cycle. The underlying genetic architecture of a trait that I'll call yield, but it's a fictitious trait, but it's one trait, it's controlled by 100 genes. I'll allow with 2 alleles, I'll allow these alleles to segregate in the population that I'm selecting within, and they're going to have an underlying additive effect, which if you go to Falconer and Mackay, is the contrast of the homozygous states for a particular gene that influences this endpoint trait, is just the average of the difference in those values or the deviation from some midpoint reference point. So we're talking about these deviations here, which we talk about as additive effects. If a heterozygous individual deviates from the midpoint, you'll hear people talking about a dominance effect as well. But I'm sort of going to ignore that for purposes of our discussion here. And I'm going to just say these 100 genes all have equal effects, which is sort of the simplest scenario, or they could all be drawn from some underlying singular, simple normal distribution, values drawn from a consistent distribution. And they'll all start in this population with a frequency of around about 0.1. And with simulation, we can take a population of 1,000 individuals in this scenario, do 100 replicates of the breeding program. I'm going to run it for 30 cycles. And if this all worked for one cycle, we should see a shift in the mean of that population. And the difference in the mean of these 2 populations would be what we're trying to predict as this genetic gain. Okay, so run that. It's on a computer today. This would take sort of a couple of seconds at most. And what you can actually get is an output that allows you to track the improvement in overall mean of the population. This is cumulative response, the top left here. We can actually look at how the heritability component that is the ratio of the genetic variance to the phenotypic variance for the trait changes over the cycles of selection, and the underlying genetic component, how that changes over time. And down here, I've just looked at cumulatively, looked at the distribution of how each of the favorable Q alleles for each of the 100 genes over the 100 replicates over the 30 cycles, the distribution of those, the frequency changes that underlies this change in the overall mean response and these changes in variance. So, I mean, if you look at it from a mean perspective, in this scenario, it sort of pretty much plays out as we would expect. And you can see this difference in mean for one cycle is equivalent to this single increment here. And then if that continued over time, we would expect a long-term improvement in mean. So this picture is sort of somewhat reminiscent of the long-term gain that you see for yield of maize from the long-term breeding of maize by the pioneer breeding program starting in the 1920s, 1930s through to present day. There's sort of an interesting way to think about this in that we can look at these overall responses and it all looks pretty predictable over the 100 replicates for the 30 cycles. And the genetic variance largely changes as we would expect. One thing that sort of is always challenging and really interesting to me, and this now starts to tie into some of the challenging questions related to trait genetics, is that all of these genes, all of these alleles have exactly the same contribution. They're exactly the same effect on this endpoint trait, but you can see that there are many possible trajectories of how the favorable alleles for each of these genes were changed over time. And it's sort of pretty interesting to me that two different alleles that have exactly the same potential at the start could take trajectories as different as this green trajectory here, which went from the low value of 0.1 to fixation, to another allele, which had exactly the same potential at the beginning, which didn't move for about 20 cycles, then all of a sudden slowly started to go up. There's nothing in terms of the gene to phenotype relationship that differs between these two alleles. The overall most common trajectory is this underlying modality under here, which does underpin a lot of what we actually see here, but really starts to become tricky to work out what's happening when you start to look at the individual genes rather than looking at this overall phenotype. And I think I'm just sort of trying to present this as some of the challenges that we actually face when we're trying to understand the genes that control traits and how we might be able to drive a breeding program by selecting on specific genes. So moving on, so that was a nice simulation example. And so what I want to do is now start to build up to a real breeding program. Life is not as simple as this. One of the key things that will be discussed as I progress through the talk is that underlying this gene to phenotype relationship and in the breeder's equation, this additive genetic variance is this little thing called alpha here, which is in a quantitative geneticist's terminology is the average effect of allele substitution, where you're swapping one favorable allele for another allele inside the reference population of the breeding program. And in that simulation example that I showed you, for each of those genes, this average effect of allele substitution was constant. It never changed over the history of the breeding program, which I think most people would consider to be unrealistic, but that's sort of the underlying basic infinitesimal model principle that's used in a lot of quantitative genetics models. So now we've got a reference population of genotypes. We've actually got a more heterogeneous target population of environments that we actually have to sample. And we don't do that by just taking one environment. We usually sample many environments by testing genotypes at multiple locations and years in what we might refer to as multi-environment trials. And then samples as selections from a potential population, a reference population of genotypes, are tested in these multi-environment trials. And in the standard selection on phenotypes, we would analyze a lot of the empirical data that came out of these experiments, and then we would actually make the truncation selection. And so what we would expect then is that by crossing the selected individuals and reconstituting the next cycle, we would get this underlying change shift in the mean of the population, so the individuals would be improved over cycles of breeding. So it's all, I mean, breeding, by and large, there are many studies that have been done on multiple crops that, yes, breeding programs, if they're run correctly, can drive this long-term gain with these incremental improvements. And obviously the question we're asking here is, can we actually do better by thinking a little bit differently about trait genetic architecture? So one of the things that has changed quite dramatically for us is that certainly in the commercial world, any of the individuals that we're creating in a breeding program and testing, we can now fingerprint quite densely with markers such as SNPs across the whole genome. And we can actually organize those SNPs into clustered groups that we refer to as haplotypes. And we can actually trace these haplotype blocks over the history of breeding. And so this example down here is just from a small segment of a chromosome, where along the top we have different individuals, and we've color-coded the different alleles from some of the ancestral founders with different colors, just to show how the different founders have contributed to these individuals in this region of the genome over the history of the program. So you can think of these allele contrasts now in terms of these color contrasts. So the question I'm really interested in is, we can do this, we've got phenotypes, and so can we do better in terms of building these gene to phenotype models by improving our phenotyping capacities using combinations of field-based testing, controlled environment testing, which I'll spend a bit more time talking about, and using a crop model as an integrative framework when we're dealing with multiple traits. So that's sort of my view of the breeder's equation in action. And I'll just pause there and just ask if there are any questions about that setup. No? Okay, I'll keep going. So here we have our basic model, and we have our polymorphism at the sequence level. We've got SNPs, and we have phenotypes. We can now implement a number of mixed linear modeling approaches. This is one that is quite robust and has been used quite heavily, Bayes-A. There are other GBLUP, genomic BLUP methodologies that would work similarly, where we're actually trying to model across the whole genome using the SNPs, the effects of the SNP alleles. So you can see this UK here ties to these effects that we're capturing in these average effects of allele substitution. The Z is a matrix of these allele scores for all of the individuals inside our experiments. So what we want to fit is basically a relatively simple regression type model with an intercept for the variation for the traits. So we're trying to get SNPs that have a linkage association with these underlying genes that we can then associate through this methodology with the variation for the phenotypes that we measure here. So it sort of seems pretty crude to most people when they start to think about what we're doing, but it works relatively well. And so here's an example from commercial maize experiments for a range of traits with different heritability levels, so high heritability traits to low heritability traits. And here we're predicting from 1 year to the next. And this is the type of predictive correlation that we actually could get here. So what this tells me is that I can predict to next year the expected performance of new genotypes in new environments with their SNP profile using this fitted regression model to the phenotypic variation in some training data set for any trait that I'm actually working with. And that's sort of the range of values that we typically see for maize in different stages, but this is in one particular stage. I'm just going to go back to this picture here and just emphasize that one of the things that we're really interested in these multi-environment trials is using these data that we've actually collected as the training data set, where we then have fingerprints of many other genotypes that were never actually tested, so that we're now looking at this question of how we predict new genotypes into new environments. So with these predictive accuracies, we can use genomic prediction to drive breeding programs where we can upscale the size of our breeding program by fingerprinting many more individuals than we can actually field phenotype. And today, the cost of genotyping is orders of magnitude cheaper than the cost of a full field plot evaluation of these genotypes. So that sort of again is now sort of bringing us back to this context. How can we use our understanding of the traits that might be contributing to yield variation in environments that are limited by water stress, water deficit and causing water stress relative to typical Corn Belt environments or very favorable environments. And obviously we want to be able to continue the ability to improve potential yield and also performance under drought conditions whenever they turn up. And I'm going to spend a bit more time just explaining how we do that using the crop growth model. And so going from this nice cartoon that shows that there's pretty basic inputs that you might think about, radiation, water through precipitation or irrigation, fertilizer, nutrients. So you've sort of got radiation, water and nitrogen on this picture driving growth and development, partitioning and ultimately yield. Obviously, we have to replace that cartoon with something that's more formal. So going down to the next level, we can represent that as a flowchart where you have these environmental inputs, radiation, water in here, and also temperature and the vapor pressure deficit of the environment influencing the balance of the conditions in terms of the radiation to drive growth and development, water availability and water deficits that may limit the ability to realize the full growth potential when water is not limited. And that drives through biomass accumulation, so capturing water radiation to drive biomass accumulation through concepts in crop physiology such as radiation use efficiency, transpiration efficiency to produce biomass, and then partitioning into grain yield. And typically, a lot of crop models are developed for only a few genotypes. And so what we were interested in doing was really extending this framework to allow us to do genetics of the components inside these crop growth models. And so here's an example of some of the traits that we've looked at that relate to reproductive resilience. So in maize, if you're familiar with this, you have the tassel releasing pollen. In the ear, the silks are exerted. The pollen has to fertilize the silks to set a kernel. And the separation of those is often referred to as anthesis silking interval, which is related to reproductive resilience. So this is this part here. Canopy size with the number and size of leaves, radiation use efficiency, and this will tie back to the limited transpiration efficiency I'll talk a little bit more about as an example. Well, we need to go beyond this sort of line diagram, and each one of these arrows can be replaced by an actual functional equation that allows us to then start to think about, is there genetic variation for any of these coefficients that are underlying the equations that are inside this crop growth model? And so I'm going to pick an example just to show you the types of experiments that we'd actually do. Brand introduced this idea of water balance and the transpiration efficiency and limited transpiration. So I'm going to talk about this breakpoint trait that we got very interested in, which came out of a collaboration with Tom Sinclair and work where he had previously studied soybean, and we wanted to see if this was also relevant in maize, where basically the hypothesis was that as your vapor pressure deficit or the dryness of your atmosphere increased, the demand on water through the stomata would drive up transpiration. And if there was some limitation on the ability to transpire, in the high vapor pressure deficit environments, with differences in this limitation on transpiration, you would get differences in the amount of water transpired. And throughout the course of a day, that varies in the amount of the vapor pressure deficit with different breakpoints on limited transpiration. You might save incrementally different amounts of water during vegetative development that could be available during reproductive development and grain filling. So we took this hypothesis and started to run a whole lot of experiments and screening. To cut a very long story short, we indeed found that with Tom Sinclair's screening system, there was variation for this breakpoint in elite maize germplasm in breeding programs, and certainly in the breeding program that I was running. And so then we started to run experiments to evaluate that in controlled environments right through to the field. And about a decade of work took us to this point where Charlie Messina and his team could actually build some of these chambers with appropriate sensors built into them to measure gas exchange at a canopy level to say, well, does all this translate into differences in amount of water that's being transpired at the canopy level in the field, and can we measure genetic differences for that? And so using this chamber system here, here's an example where we compared something, hybrid that did not have the limited transpiration with the open circles where one that did. And here are the differences of transpiration in one particular day, looking in a high VPD type environment, just looking at the difference during the day, and you get this significant difference in the amount of transpiration that was actually occurring in the course of that day. The next question you could ask is, well, does that matter? And so does that matter in terms of measurable differences in the amount of water in the soil? So we had to improve our methods for actually measuring soil water extraction by different genotypes. And again, after a long history of developing different technologies and working with different technologies to do this, we got to the point where we were quite comfortable in being able to measure in the order of 5 to 10 millimeter differences in cumulative amount of water use of hybrids over the course of a season. And here's an example from an experiment that complements what we're looking at here, where in this case, the hybrid that is the open circles in this is actually the one that did not have the limited transpiration. And you can see that what's actually happening is it's depleting the soil water at a greater rate than over the course of time than the hybrid that actually had the limited transpiration. So there's more soil water available in the soil profile. And this difference at flowering time translated into about a 25 millimeter difference in available water at flowering time in this particular experiment. So then the question you can ask is, does that matter? And so in these experiments where we could manage water inputs and we had high vapor pressure deficit, we could show that for these types of hybrids with this difference, that we could get a difference in yield of these hybrids, where this hybrid in this case completely failed in this plot. whereas here we still managed to get about a 10 ton per hectare yield for this particular hybrid in this experiment, in this situation, in this plot comparison. So we then took this back, and with the crop growth model, we could start to look at this over the wider range of environments across the whole Corn Belt. And you can see that the extreme that we're having this experiment is the extreme situation, where there's about an 8 ton per hectare difference here. But in a lot of cases, there are much smaller differences. But in a water deficit environment, we actually saw that there was generally a benefit. And the important thing was there wasn't a huge cost in the more favorable environment. So we could actually manipulate by selecting appropriately for the right breakpoint, so that we weren't costing our yield potential, but we were actually building in more insurance and through these different combinations of water saving approaches to give us yield like an insurance policy in these water limited environments. So that's sort of the background. So it sort of then helps you to start to think about the differences in approaches that we could start to think about in terms of building up our phenotyping data sets. So we could do a whole lot of testing across the Corn Belt. So 2012 was quite a famous drought year in the US Corn Belt. And here, the red, so each point is an experiment. And in these experiments, we've just identified those experiments where there was a water deficit at flowering time. So these sort of two solid lines and the dashed line down here at flowering time. And we've highlighted those as water deficits that are causing drought at flowering time or into grain filling, whereas the green, there was no other limitation, but so they were more like the favorable environment. So there wasn't a water deficit. So you could do this coarse grain comparison between drought and favorable environments throughout the Corn Belt with our environmental characterization capability. And you could start to break out these data sets to do the training of our genomic prediction. Or we actually designed specific stations where I could run all of the stages of my breeding program in different levels of water deficit. So from severe water deficit through to more favorable conditions representative of high rainfall environments in the Corn Belt, so that we could stratify the types of environments that we were getting through wide area testing at one location. And this is the research station just south of Santiago, Veluco, where there's basically no rainfall during the course of the season. So we manage all the water inputs through drip tape irrigation, and we get high VPD conditions. So this experiment was also run there, and this type of testing was done there. We do other strange things inside different types of controlled environment facilities, where we look at how this could translate into differences in silk exertion. So here we're just attaching different transducers to silks and different parts of the plant to measure silk elongation rate. And we can actually measure how this translates into effects on silk exertion and ultimately kernel set, and also use that inside the crop model. So what we then wanted to do was to take this basic idea that I discussed earlier, where we were doing genomic prediction for endpoint traits, but we wanted to point that at traits that were coefficients inside the crop growth model. So basically what we're now doing is we're embedding the crop growth model inside the genomic prediction algorithms as part of the link function. And inside that crop growth model, we have particular traits. So instead of doing the genomic prediction to the endpoint trait yield, we're actually doing the genomic prediction to these traits inside the crop growth model. And then we're using the crop growth model to integrate our modeling of the traits to the endpoint yield. And to do that, we basically do that within a hierarchical Bayesian modeling framework, where we can sample the marker associations to the traits with a Gibbs sampler, and we can sample the predictions from the crop growth model using a Metropolis Hastings algorithm. so that we can actually build posterior predictions for yield and also for the underlying traits. So this is just the methodological framework for putting that into place. So what we've now done is we are taking the crop growth model, we're identifying coefficients inside here that are traits of interest. We actually use our phenotyping to build the priors that actually underlie the components inside the Bayesian framework. We sample from those for the underlying traits and then use the crop growth model to predict yield. So it becomes a very different phenotyping strategy where we can actually phenotype to build priors rather than having to phenotype empirically to phenotype every individual. So our framework is sort of really an integrated phenotyping around this methodological framework to allow us to predict yield outcomes. So if you want to see it in a cartoon that's similar to the example that I gave you before, instead of pointing our genes to the endpoint traits, we're actually pointing them to traits and then using the crop growth model to integrate their effects. The important thing that I won't go into a lot of detail here about is that this then allows us to deal with the fact that G by E interactions or trait by trait interactions can actually give rise to changes in this average effect of allele substitution. And this is where we can actually potentially get this lift in our ability to get this predictive accuracy. And so what I want to do now is just go through some examples to show you empirical evidence that motivates us that this is actually working. So this is my breeding program, some of the early stages of this. So there are actually populations being tested under well-watered conditions and different types of drought at the same station. And so what we can do is we've got genotypes, they're all fingerprinted. We measure yield, so we can actually do the standard Bayes type genomic prediction on yield using these data. Or alternatively, what we can actually do is use the crop growth model. And here, what I'm showing you now is a comparison for one of these data sets for 4 families, so about 350 hybrids, with around about 100 hybrids from each of the 4 families, a little bit less than that on average, obviously, and predicting from the well-watered into the drought and from the drought into the well-watered environment, and predicting one of the families based on training in 3 of the other families. So we're trying to predict new genotypes in new environments. And here are the different combinations for these 4 families by 2. So there are 8 points on here. And if you remember this graphic, this is the standard Bayes prediction on yield with the same data set. This is what we could achieve with the crop growth model. So we could actually get this lift in more situations than we lost the lift of this predictive accuracy. And this sort of then enables us to drive the outcomes from genomic selection harder than if all we're doing is looking at these endpoint traits. So this work, this empirical work, is actually published in an article where Charlie Messina is the lead author in the European Journal of Agronomy as part of a special issue from the International Crop Modelling Conference, the first of those. So a more recent publication that I think I could point people to is available now on bioRxiv by Christine Dijpenbroek, Tom Tang, who worked with Charlie and myself. Now we're scaling this up to a much larger full breeding program level where we're looking at like 35 families, about 2,367 hybrids. So this is only on one side of the heterotic group in about 23 environments. And basically what we can show you again is that in some of these situations, we're getting this predictive accuracy lift. In some situations, we were not. But if we look at a lot of the different within population predictions, you can see that there are a lot of situations where we were starting to get this lift. And I'm going to sort of condense down the story to let you see how we've interpreted that. So we've now organized all of these data so that we can actually look at where we're getting that predictive accuracy in terms of the level of drought that occurred in the situation that we were actually looking at, the particular environments. And like in the previous example, but on a larger scale, for those environments where we were actually getting more severe stress was where we were able to better deal with changes in average effects of allele substitution due to G by E interaction effects and improve our predictive accuracy using the crop growth model over just predicting the endpoint yield. So this is the difference in predictive accuracy between the two methods that we're actually looking at here. And again, we didn't lose too much when we went up into these highly favorable environments. So I'm just going to sort of wrap up now with a couple of summary slides. And what I'm going to do is just go back to some deep theory that I did without going into the background behind this. But this was a theoretical prediction that we actually made from modeling many different G to P architectures of traits and how they influenced quantitative trait outcomes, where we were comparing different molecular breeding strategies to phenotypic selection. And what we could show underlying these long-term responses was that for some of these more complex situations, so each one of these points here is a different genetic model underlying a trait, like the empirical examples that I showed you before. So these are inspired by empirical data, but they're all simulated examples. And we've organized the scenarios from the more complex to the less complex to the more complex. And as you saw of the theme that I was trying to show in the empirical results, what we actually saw was that these additional advantages we were getting from using the integrated phenotyping and crop growth model were predicted to happen in the more complex situations back here. This was before we ran any empirical experiments. So we then sort of started to hypothesize that maybe in these more complex traits that we're working with, such as drought tolerance, instead of just trying to select for yield in drought tolerance, there may be some opportunities for investing in building this greater gene to phenotype knowledge to build a framework for enabling enhancements in our genomic prediction strategy. So this theory said there were advantages. The challenge, though, was for us to come up with a methodology that allowed us to capture some of these features of this gene to phenotype relationship and to enable the prediction. So we had to embark on an appropriate infrastructure and capability to build appropriate gene to phenotype models. I've sort of tried to cover some of the background behind that and sort of talked a little bit about some of these empirical examples. So in terms of sort of matching the three parts together, you've sort of got this deep theory in quantitative genetics, which I haven't really gone into, but this underpinned why we were embarking down this approach, the investments in building our prediction methodology framework beyond the standard genomic prediction, improving our phenotyping strategies, improving the crop models and integrating those. And then sort of here's our first real view of this complexity continuum, but now related to a drought scenario in that experiment that I described, where we're actually getting some preliminary empirical evidence that in some of these more complex situations that we may indeed be able to deal better with changes in average effects of allele substitution and make better predictions. So that sort of wraps up the contents, just a quick list of current themes and future directions. There's ongoing work to continue to develop the methodology for maize applications. Maize working with the multinational situations such as Pioneer, Corteva is sort of an advantage, is data rich, and there's a lot of opportunities. So that collaboration continues. We're interested in the opportunity to transfer this capability to many other crops. So that's what I'm sort of really focusing on now. So one of the model systems we're working on is our ability to transfer this to sorghum. So that's work that Owen Powell is leading as a postdoc with me, and working with David Jordan, Graham Hammer, and Charlie Messina, and others. We want to keep advancing the theory and methodology, because I think that what I've shown you, while it's encouraging, it's still very early days from my perspective, but I think that there's enough interesting results from the theory and the empirical work that it seems worthwhile developing this a bit further and thinking a bit harder about a whole lot of things about selection, applications to crop breeding, and selection theory as it applies to natural systems. And that's the connection with Daniel Ortiz Barrientos. And we're really interested in, some of the areas that are quite interesting to us are how we would perhaps merge some of the ideas around using these mechanistic crop growth models with a lot of the big data ideas from machine learning, artificial intelligence algorithms. So we're sort of interested on that space. And I think an underlying theme now that I think we've sort of cracked the door open is how to use prior knowledge of gene networks and traits, embedding that in the framework and the methodology that's used in breeding programs and actual breeding programs to enhance prediction. And that's a core theme of the Center of Excellence for Plant Success that Christine Beveridge is leading, that I'm participating in. So with that, I think I will stop there and say thank you for your attention. I'm sort of anticipating that you're all asleep or nobody has any questions, but I'll put up there the invitation for questions now. And my favorite cartoon, I hope I did a little bit better than just showing alpha, and it seems like then a miracle occurs still, but by trying to explain the gene to phenotype relationships that we're interested in to better use our knowledge that we have about traits and how they contribute to things like yield and yield stability. So I'll stop there, I'll stop sharing, and I'll make sure that, oh, Brandy, you're still there. I'm glad to see that because I couldn't really tell, but I'll hand it back to you.
Thank you very, very much, Mark, for that very impressive tour de force of plant breeding and how to apply it to an important crop and important trait. I've got loads of questions, but I'm going to bite my tongue because we'd like to encourage the students and the postdocs to ask the first questions. And I'm glad to see that there's a couple of people who've raised their hands. So Haneen, can you unmute yourself and ask the first question, please?
Yes, thank you so much for your talk. I guess I have two questions. So one question is, what do you think what are the factors that actually can affect your genome protection models? Because given the fact that there is a accumulation of deleterious allele, gene flows, polygeneity, and these can actually affect the phenotype, and when you want to do the phenotype-genotype prediction. So how do you take into account these facts in, for example, the yield genomic prediction or any phenotype to do the genomic prediction? This is my first question. Another question is, if in the first slides when you were speaking about how you do this genomic prediction, you talked about that you have your SNP matrix and then you do the haplotype from this SNP matrix. How about the structural variance in genomic prediction models? Because given that there are so many phenotypes that are linked, or that there are structural variants that are linked to phenotype. So do you take into account structural variance in your genomic prediction models, or it is a little bit advanced that it needs more things to do until we reach the level of doing that?
Two big questions that could then generate a lot of discussion. So what I'll do is I'll start with your second question first, and then maybe go back to the bigger question. So the structure and the way that you may be asking this is sort of like in the classical GWAS, we take into account the structure of the germplasm and the relationships. Is that the type of structure you're talking about?
No, genomic structural variance is not just a single nucleotide polymorphism, but yeah, like big deletion insertion.
So you're talking about the underlying LD structure that exists across the genome. Is that what you're asking about?
Big structural variants like insertion and deletions and so on, because these structural variants are linked to phenotypes such as colors, taste, and so on.
Yep. No, absolutely. And so, as you know, maize is sort of a really interesting system to study structural variation. There are indels all over the genome. The SNPs that we're using in the empirical examples that I showed you there were not associated with those structural variations, but they're distributed throughout the genome. So these are SNP polymorphisms on sort of standard, where you have surrounding sequence where you can assay an actual SNP, a true single nucleotide polymorphism. So that sort of works even with that structural variation, which does exist inside the variation that exists in these breeding populations. So that works. That being said, there's another component of variation that's not accounted for by those SNPs that could be as easily targeted by looking at that structural variation as well. So basically, the mathematical framework and the theory can totally handle that. You could define structural variants as the polymorphisms, and you could then associate that as another class of molecular information inside this prediction framework as well. So there's nothing that actually stops you from making that explicit within inside these prediction frameworks. It may reveal additional information that's not captured well by the standard SNP panels that I was using in this empirical example. So, and the important thing is, as you would know, the SNP variation that we're capturing is not all of that variation, and another part of it may be explained by other sources of genomic characterization that were not used in those empirical examples. And I totally expect it will be from our knowledge of elite maize germplasm. So then the next point about your gene flow and all these other things, the specific examples that I talked about here are elite breeding populations that have undergone many cycles of selection. So in terms of looking at the material that I'm studying here, you're looking at the endpoints of many cycles of selection. So that's the type of standing variation in these empirical examples. But again, being said, without making it much longer than it has to be, because you could have a whole seminar series on this, these are topics that we're looking at in the Center of Excellence for Plant Success, where we're wanting to go right across the pangenome that goes across many relatives and look at how evolutionary trajectories may actually tell us more information, prior information, that we could better utilize when we go into breeding programs. And that's the connection between the natural systems and the breeding programs. But at risk of talking for too long, I'll just leave it at that and maybe go back to Brand. Hopefully I answered at least partly your questions. And it's an invitation. If you want to ask more, you can always email me.
Yeah, thank you. Thank you so much.
You're muted, Brand.
I was muted, sorry. Nico, you've got your hand up. Thank you.
Yes. Thank you so much for a great insight into large-scale breeding that we don't often get here. I wanted to ask you about your opinion on the current and future effectiveness of breeding by selection, because, well, this is not really my field, but I imagine after 100 years of breeding that the low-hanging fruit might be gone, and that it's smaller and smaller increments of change that you can achieve this way, and that it becomes harder and harder to measure above the noise. And maybe also that the traits that could see significant improvement are more complex, in the sense that sometimes you first have to go to lower yield to get higher yield later, or such structures. Do you see that as a limitation currently to breeding by selection, or soon in the future? Or, well, you've given a few examples of how to sort of sidestep the issue and extend the effectiveness of this method. But I was interested in your opinion on this issue. It seems like an arms race.
Yeah, no, they're really good questions. Well, the collection of things that you've opened up there are really good, Nico. So, one of the things, when I joined Pioneer, I collaborated with them in the 1990s and then joined them full-time in 2000. And part of the reason I joined full-time was because I thought, so I started off as a wheat breeder, and I sort of was always interested in maize because I thought, well, maize must have pretty much exhausted variation, and therefore there's no real opportunity for further improvements. But it was sort of surprising to me when we actually started to look at the phenotypic variation for some of these traits, and at the sequence level, that there's an immense amount of diversity at the sequence and at the phenotypic level. And I think most people, when they start off looking at maize across the Corn Belt, they think it's all the same. It's completely uniform. But when you actually start to look at it, I'm amazed at how much diversity is actually left and what's still to be sorted out. That being said, I think that there's two types of questions that we have to ask. One is when you're breeding by selection, to actually remove weaknesses from hybrids, or varieties that are already being used in agriculture. If you look across a lot of the widespread use in a lot of the major crops, there's a lot of weaknesses as you go across many environments. So I think that in terms of improving stability, I think in terms of on-farm productivity, even if we don't improve that potential, there are better ways to improve stability than what we've been able to do in the past. And that will translate into less yield gaps on farm and better sustainability and productivity, even if you're not moving that potential higher. And then you can actually, instead of confusing all of these questions, you can focus on that stability breeding, which was what the Aquamax breeding was. And if you wanted to improve the potential for yield in favorable environments and the potential for drought tolerance, you can start to focus on different types of traits. So it allows you to target when you're actually looking for novel diversity versus selection and how you bring those together. And I think that just probably opens up a whole lot of the ideas that you're already thinking about and talking about. So that's sort of where I'd leave it at that point. But it's again, it's one of these topics that I think is really important to really dive into when you're thinking about how selection and novel diversity can contribute to genetic improvement and improvements in on-farm productivity.
All right, thank you very much.
Thanks for that, Nico. Sabiha, you've got your hand up.
Yes, yeah. Hi, thanks for your presentation. I'm just curious to know that, have you included the grain quality parameters in any of your equations? Like I can see you have a grain number and the grain size. Yep. So it's like if we are talking about any of the staple crops, let's say if it's a rice, then how important it is to go in parallel with the stability as well with the grain quality?
In the maize example, we did not. So that's sort of one of the aspects about maize for its major commodity uses in the US Corn Belt. It's basically fed to either ethanol refineries or to cattle. So we didn't have to worry too much about the quality, but your point is well taken. And so in terms of extending the methodology that I talked about, I won't go into the details, but we are working on multi-trait approaches that allow us to look at yield and quality components. So it's extendable, just like selection theory is extendable into the full G matrix, where you can actually look at yield and quality traits and other agronomic traits and even specific stress tolerance traits. That can be built in, but in none of these examples have I shown you an example where we looked at quality, but it can be done.
All right, thank you.
Thanks for that, Sabiha. I think I might ask a question myself now. Mark, so I introduced you as a bit of a dark horse who started off in academia, and then you went into industry, and now you're back in academia. And as group leaders, we often get questions from, or sort of comments from students and postdocs saying, you know, I think I'd like to go into industry. And so for someone like yourself who's worked in academia, then in industry, and then gone back into academia, could you give us a little bit of a reflection in a few words about, you know, what were the perhaps advantages, what was fun and good about working in industry, and you know, why did you leave, and what are now the advantages? What freedom do you have in academia that you didn't have in industry? So for the benefit, especially of the students and the postdocs.
Yeah, no, I thoroughly enjoyed my life in, and still enjoying, I guess I should say, life in both domains. And what I would say is that it's put me in a much better situation to be able to negotiate and establish really effective public-private partnerships. And so I've worked with a lot of people globally about how to ensure you can put in place very effective public-private partnerships. There's good science that's done in both domains. There are great scientists in both domains. I think the thing that was really exciting for me in my initial academic life was that I had the freedom to explore theoretical questions that seemed nobody else was interested in at the time. But I mean, as probably most people start off, they're told, yeah, that's a really stupid idea, don't waste your time. But I thought I'd still try that. And it got to the point where there were interesting advances that we actually made that had huge impact in a lot of domains. And eventually the industry started to come to me to collaborate, saying, well, look, you know, we're interested in doing this. We don't know exactly how to do it. And you've got a capability. Would you partner with us? And so naively, I went into that partnership, and it gave me a really good insight into the private sector, and just encountered a lot of extremely great science, great people, and a lot of resources. And I think if there's one major contrast I would say is that as an academic to begin with, I spent more time than I enjoyed doing grant applications and getting knocked back because nobody liked my stupid ideas. When I went to industry, basically they said, you'll never have to worry about resources again. And yeah, when you got $100 million a year budget, yeah, you don't really have to worry so much about sort of doing some of the things that I was talking about. You won't have to write grant proposals. You won't have to spend a lot of time sort of justifying all of your expenditure. And so a lot of my life in industry was really focused around the same questions, but it was resourced very well and developed very good, effective relationships. And so now I see that there are strengths and weaknesses on both sides. And that sort of is why I'm really interested in these public-private partnerships. So our Centre of Excellence for Plant Success is well funded by the Australian government. For 7 years, we have core funding that's sort of guaranteed for that 7 years, a lot of additional funding from institutions and in-kind support. And we have industry partners. And so these partnerships are really the opportunity to do what I was doing initially, but at a higher scale with a lot more experience. And so I think you can navigate a successful career in both. They're different. You won't really know what's right for you until you try either one. And if there's one thing I would say about anybody in the early part of your career, be like a plant breeder. Don't be afraid of failure, because 99% of what I did in a breeding program was an absolute disaster. I just don't write talks about those types of things. I think you just got to go out and explore and try some things. And I think that was probably what I wish I would have done more of earlier in my career than later in my career. So I'm pretty comfortable moving around across all of those now, and have, if there's one thing I would have rather done is probably been a bit more mobile early on, but it's hard to do that and do something substantial in a breeding program, which does take a little bit of time. Long-winded answer. Sorry, Brian.
Yeah, thanks for that insight, Mark. I think that's really interesting and useful for all the students and postdocs. Mark, you've got your camera on now. You'd like? Yeah.
Not Mark, me. Okay.
Thanks.
Yeah, Mark,
I was going to pick you up on your dark horse comment, because if Mark Cooper's a dark horse, and given I've now become an entrepreneur, does that make me a dark donkey or something? Because there is a third way, it's industry, there's academia, and there's also the entrepreneurial way. But anyway, back to science. You had the plot of grain yield. That's a good question, Brandon. Very good for the students, actually. I like it. You had the plot of grain yield against time over a very long time period, and you labeled it as long-term genetic gain, but it's actually long-term yield gain. And so I'm wondering if you'd like to make a short commentary about the role of agronomy in long-term yield gain and the interaction between agronomy and genetics. Just again, it's sort of a general question for people to hear.
No, it's really important, and it underlies some of the more recent developments in delivering improvements in on-farm crop productivity. So I think that's also another really interesting background story for maize, in that historically there was the increase in density, and then the realization by the breeders to select for density tolerance, which then allowed some improvement in management approaches. One of the interesting pieces to that a lot of people don't understand is that a big shift that actually happened was standability was a big problem. I mean, a lot of these early maize hybrids could yield really well, but standability was still a problem. So a lot of the breeders really focused on standability, and into the 1950s, people were still hand harvesting corn. I mean, you got to think about it. And so once they got the lodging problem pretty much under control, with standing corn, the mechanization industry took in. So it wasn't just agronomy, it was mechanization as well. So you actually got combined harvesters, which were combined cutting and shelling harvesters. So that's your combined harvesters. And so then with that allowed investment in planters. So John Deere were actually a big catalyst behind this as well. It drove the improvements in plant population. You had to get improvements in seed quality. So all those went together. In summary, the genetic gain for yield is usually equitably split between agronomy and all
that's right. That's
yield. The experiment that I showed you is a side-by-side comparison where we actually grow the hybrids in a lot of different management scenarios from the 1930s right to the present day. And we actually give the yield in the best management of the time. So it is a combination of yield and management that we're actually doing. So we're
that's
not
experimental
penalizing
data, not historical data. I beg your pardon.
So we're not penalizing the old hybrids by
understand that.
Modern management. We're growing them under their management. So, and these are hybrids that were widely grown by the farmers, millions of acres. So it's sort of on-farm management and yield that we're trying to look at there. But it's a good point to clarify that.
Well, but thank you. That is important. So it really is genetic gain that you're plotting on that particular graph. Then could I ask a second question, because I wasn't going to, but you've got the Aquamax there. And the Aquamax, as I understand it, was to have traits which would improve its water use efficiency. or with
performance
and thus
under water limited conditions.
And thus it's drought tolerance. That's right. Yeah. So then why is it right on the line and not having a disproportionately or an unusually higher yield in the drought conditions?
It's a really good question, and it's an artifact of trying to grow these modern hybrids with these really old hybrids. And so what we realized was that to keep in the drought experiments, to keep the old hybrids alive, we're not really stressing the modern hybrids enough. And so we're not getting the same level of separation.
Okay.
So if I was to grow that experiment under the drought conditions that give rise to the separation that I showed you in the demo, because both of those were modern elite hybrids that were in that Aquamax, in that era series. So those two hybrids that had the no yield and 10 tons per hectare are in the era sequence. They're multi-million acre hybrids, but they're not separated there, because if I grow that experiment, but 75% of those hybrids all have zero yield in the older generation. So we're actually rerunning that experiment in a way that actually stresses things a bit differently to actually answer that question, because enough people have asked it that we need to demonstrate why that happens. But it's a good question.
For the modern hybrids, you've got that yields going down from 10 tons down to like 8 or something. So it's a fair old yield stress.
It's a
drought stress.
Yes.
Yep. But you just need more. Okay. All right.
Yep.
Thank you.
So it's a mixture. We tried to get stresses that were at flowering time and during grain filling as well. And so a lot of the hybrids, the modern hybrids that are really sensitive to water limitations, if they get through flowering time, they don't suffer the same level of separation.
Yep,
so the big thing about 2012 that actually devastated the Corn Belt was that the drought happened a lot earlier, because there wasn't much storage of moisture in the soil at the start of the season.
Yeah.
Had high VPD that went into the central Corn Belt and east, and therefore you had much greater separation, which doesn't always happen.
Thanks a lot.
Anyway,
Thank you. Thank you.
There's a fair bit of detail behind that, and
that's great. That's a good answer. But thank you very much, and thanks for the talk.
So we're running out of time now, but Nico has one more question. He's got his hand up, and so we'll take one more question. Nico.
Yes, thank you. I'd like to ask about hard to detect side effects of detrimental side effects of some of the traits you select. Let's say you find a trait with a bigger leaf, but in high winds it might lodge more, but you wouldn't normally test that for high winds. So, and you cannot test for every circumstance. So how do you deal with these possible hard to detect side effects?
So I mean, it's almost like I paid you to ask the question that I wanted you to ask, Nico. So while I focused on drought here, we had a similar complex trait, which was lodging and brittle snap, where the stalks snap in high storms in the western part of the Corn Belt. So just like we've invested here, and I talked about the drought genomic prediction, we did exactly the same thing for the stalk lodging and the brittle snap, so that we were actually doing genomic prediction for all those other traits as well. So a successful hybrid, that an Aquamax hybrid isn't just a drought tolerant hybrid, it has to meet all these other criteria as well. And it was actually the selection for the combination of traits that allowed those hybrids to perform extremely well. And in a publication that I wrote in Crop and Pasture Science, which I think is published in 2014, actually, there's a bit of information about the other traits like the lodging and the brittle snap, and we talked about the genomic prediction for brittle snap in that particular article. But yeah, so I mean, to be a successful product in breeding, it's not just about one trait. I mean, we don't sell individual traits. You sell products for the combination. So it's a great question, and we spent a lot of time doing multi-trait selection. As I mentioned before, I just didn't get into that level of detail to answer your question up front, but I'm glad you asked it.
Right. Thank you very much for the good questions, everyone, and especially thank you to Mark for an absolutely excellent seminar and great discussion. So I'm sure you'll like you said, you'll be happy to answer additional questions by email and so forth. So please join me all in thanking Mark for his seminar.